went to Canvas this morning, and all my passwords were down. I couldn't go to WebAdvisor, I couldn't access my email, I couldn't access Canvas. So my password expired. No one told me. So I called. Oh yeah, we expired your password. I'm like, why? So I've been fired? No one told me? You had a question?
Yeah. I'm not sure if you answered, but so when parents pass down a gene that's been silenced,
Yep.
can the child turn it on, or does it stay silent?
Usually, well, imagine like in the classic example I give you on the insulin growth factor 2. If you inherit the paternal version, it's activated. The maternal version, no matter what the gene says, is never used. Okay. Now, if that comes to you, now you're not a male. So when you go to make eggs, you're going to remove that silencing. So you're well, no, I'm sorry. So the male version is active, the female is masked. Okay. So if you inherit an active male version, you will, when you make eggs, you'll remove that silencing. Let me write this down because I'm going to confuse myself. Paternal and some imprinting, you can do both. They're both examples. And let's say the paternal one is off. If you inherit the gene, they move from the paternal side, it's always off. And the maternal side is on. Okay. Okay. So we got a male and a female here. Okay. When it passes genes to the offspring, okay, these are going to be off. These are going to be on. Okay. But let's say this ends up in let's say we have a daughter here. Well, this daughter now, when it gets these genes, she's going to be able to kind of reactivate that paternals back on. So every time we make sperm and egg, we reset them according to the gender. So it is reset. Okay. That's the point I was trying to make. All right. We're going to jump into chapter 14 here. And this chapter starts off with kind of a introduction into how DNA was discovered as the molecule of inheritance, because for more than half the 20th century, DNA was not thought to be the molecule of genes. Okay, and let's see why that was not an illogical point of view. So by the beginning of the 20th century, according to Walter Sutton's theory of chromosomal inheritance, chromosomes are the carriers of Mendel's particles. But it was known that these chromosomes were composed of two types of molecules, proteins and nucleic acids. So which component of the chromosome carries the molecular information? Okay? We know that Morgan's lab confirmed the chromosomes were the carriers of the particles. But when you see a chromosome, which is half protein, half nucleic acid, 50% by weight each, which molecule carries the genetic information? Well, most scientists at the time believed that proteins were the molecules of inheritance. Why? Because you can imagine a molecular code must be a very complex code. If you're going to code for all traits and all living things, that must be one hell of a complex code. And proteins are complex molecules. They have an alphabet of 20 amino acids, where nucleic acids are relatively simple in terms of their structure. They too are polymers, but their alphabet's very simple relative to a protein. So it just seemed logical, based on this difference in complexity between proteins and nucleic acids, that proteins must carry the molecular information. And that a nucleic acid, maybe it's just like a support scaffold to maintain the protein structure. That was the prevailing thought through much of the 20th century, at least into the 1940s. Make sense? So that's a logical argument, right? Okay? Well, here's the first experiment that began to push the focus toward DNA, the molecule of inheritance. Performed in 1928 by this fellow here with the dog, Dr. Frederick Griffiths, a British medical officer who was studying. He was later killed by a German bomb in the Blitz in 1940, unfortunately, but that's 12 years later. He was studying, he was a British medical officer, and he's studying pneumonia. You want to keep the soldiers healthy until they get killed, right? So he was studying two phenotypes of pneumonia. This is caused by a bacteria that's called Streptococcus. This is its modern name. Pneumoniae. I think I spelled it right. And it comes in two phenotypes, or what most microbiologists call strains. Okay? One is called the R strain, Here's a photograph of these strains and the S strain. This is now bacterial colonies growing on a culture plate. We're going to culture bacteria next week in lab. Okay? Where you inoculate an agar plate that has, an agar just gives you a gel support. It's impregnated with nutrients to facilitate bacterial growth. You can change the nutrients to favor certain bacteria. But if you streak the bacteria in the agar plate and let the plate incubate, the bacteria grow into large visible colonies because these bacteria divide over and over, and one cell produces a patch of cells that we call a colony. And you can see the R strain, the colony sizes are much smaller. S strain is larger. That's because the S strain, they produce a gelatinous capsule around the cells. You can see the capsule here is in gray. And Griffith named the S strain S because this capsule tended to make the colony look smooth on the edge, the periphery of the colony. Had a smooth texture to it. The other strain, the R strain, these are cells that the rough strain or phenotype, these cells did not produce a gel-like capsule outside the cell, and the colonies on the agar plate visually had a rough texture to them. So we had two strains, smooth and rough. Okay? We could skip this for right now. We can come back to that, talk about that in a second. Let's look at his protocol here. Now, what's interesting is that these two strains, one causes disease that is pathogenic, one is not. So, of course, you can't inject, even in the military, you can't inject soldiers usually without their permission. So he's using as his lab animal a mouse, which is a good model organism to study disease, since we're all mammals. So I hope you can read this here. So what Griffiths would do is would make injections of these two different strains into mice and look for the pathology. And so when he injected living S cells into a mouse, the S cells with the gel capsule caused pneumonia. The mouse died. And then when he would autopsy the mouse and plate the fluids on an agar plate, the S cells would grow. That makes sense, right? You inject with S cells, they kill the mouse, and then the descendants of those cells, their descendant cells are S cells. Okay, I get that. So the S strain is pathogenic or virulent. Okay? Make sense? It's very straightforward, right? When he had the other strain, the R strain that lacked a gel capsule, when he injected these into the mouse lives. The R cells don't even survive. They're killed by the immune system. Okay? So there's no disease, there's no pathogenicity, they're non-virulent. So evidently the capsule is responsible for the virulence of the S strain, which it is. The S capsule tends to protect bacteria from attack by the immune system. Doesn't mean it's going to necessarily be toxic, but if these cells produce some sort of a toxin, okay, they're protected by the immune attack by the capsule that the R cells lack. Okay, all right, so no big deal. Now, as a control, to make sure that just the injection of the S cells itself weren't killing the mice, he took S cells, living S cells, and he heat killed them. So he could inject an intact capsule, but the cell is dead. You inject that into the mouse, and of course, the mouse lives. He's injecting a dead cell, okay? And no live cells are recovered on autopsy, okay? So living S cells are pathogenic, and we recover the dead mouse and collect body fluids and culture those fluids. You produce the living S cell descendants of these cells. R cells, when they're injected live, there's no disease. The immune system kills the R cells. There's no bacteria recovered, no descendants. When you heat kill these cells, gently heat kill them, so you kill the cells with the capsule's intact to make sure the capsule itself is not pathogenic. As a control, well, it's not. It just protects the cells. Mouse lives, and there's no living cells, so you're not going to recover any living cells. That makes sense, right? Everything's sensible so far. But here was the weird part that Griffith had to interpret. He did a dual injection where he mixed together the living R cells that are non-pathogenic and are killed by the immune system with the heat-killed S cells that are non-pathogenic and they're dead already. The friggin' mouse dies. Huh? And what you recover are living S cells. Is this like a reincarnation? Is this a second coming? Should I get religious here? Do I need a priest? No. Never. Okay? Never in science. I'm not saying priests are bad, except for us, they are. inheritance only with your son. Okay. Exactly. All right. Anybody got a brother that's a priest? Is he in jail? Just kidding. He should be. No. So think about this. Sorry. I hope any Catholics in the room, I didn't mean to offend you. I'll offend all religious groups equally. Yeah, there you go. There you go. I'm an equal opportunity offender. Okay. So when you do the dual injection, individually, the living R cells are non-pathogenic and they don't survive the injection. The heat-killed S cells, they're non-pathogenic, and of course they're already dead. They're dead. When you do the dual injection, the mouse dies of the disease, and the cells you recover are the living S cells. And when you allow them to reproduce, their descendants are S cells. They've been genetically transformed. It's like hanging out with a blonde, next thing you know, you're dumb. You're blonde. Sorry. I used to be blonde. Still dumb, but okay. So there's a genetic transformation. These cells have transformed into the S cells, and their descendants have this new trait. They've been genetically transformed. They've acquired a particle of inheritance that's allowed them to transform genetically that they now can pass on to their offspring. That's how they interpreted that correctly. And he called it transformation. Transformation, according to Griffith, is the uptake of foreign DNA by a cell, okay, or an organism. Let's say an organism. In this case, it's a cell. And the recipient organism acquires these new traits according to the DNA it inherited. You're transformed.